In our body, there are many proteins. So if you want to study the whole protein together as a group, we call that study as a proteomics. So proteome is a whole collection of available all the proteins. So if you break up the one cell and then isolate those proteins, most likely you utilize this kind of chromatographic separations. This is the SDS gel stained with a Kumasi blue, this is blue dye. And then each lane, you can load those samples on top, and then they flow down using electrophoresis. So this one usually separated by size of those protein, and easily you can see tens of hundreds of thousands of different bands here. If it is a simpler sample, maybe you can do this kind of 1D gel, but whole proteome may have huge numbers of different proteins, then maybe this 1D separation may not be enough, and many proteins may be overlapped inside here. So then another technique you can use is maybe two-dimensional gel. So here you can do running one gel first and then running the other direction. So usually the parameter you can utilize is similar to the 1D gel. One is along line the size of those protein. The other one is the pH. Isoelectric at the point of different protein. Then you can separate to two different dimension of protein separations you can do. If a proteome is a whole collection of proteins, how many proteins in a proteome do you think? When we sequenced the whole human genome, we found that we may have a little more than 20,000 genes. 20,000 different information for 20,000 different proteins. I said that number is surprisingly lower than we expected because we believe that our body is much more complicated. That's why we may need more diverse machineries. Actually, the situation is not so simple. So let's say we have a little more than 20,000 different other genes in our genome. When it is described into RNA, even though we just count only mRNA, that's what we call maybe conventional transcriptome, still the number is increased a lot. So many things can happen here. Maybe different promoters, they bind to the different area of a starting point of RNA synthesis. That's one possibility. And alternative splicing, we have 3 units of genetic code to make one protein. Depending on those location or space position, actually we have 3 options to make different proteins. So that's what we call the alternative splicing. And also, during the maturation of an mRNA, the intane combination can be different. Maybe all the inteins can be removed, or partially some of them remain, then it can generate a lot more complicated product of those mRNA. So that's why utilizing this kind of different combinations of options, even though the gene number is only 20K, maybe transcriptome can be much more complicated. Maybe let's say 100,000 of different transcriptome also possible to be generated. More complicated. After this protein is synthesized, now there is another possibility of diversification, so-called post-translational modification. Even though one protein is synthesized, now that protein can be modified by many different ways. Now the whole proteome number, maybe it's more than million. So now the situation is much more complicated than before. So even though we have very relatively small number of genome, but proteome number is much bigger. Now we need to understand not only the sequence of this genome, we need to understand what kind of changes happened to this protein. Let's say this is synthesized, the first product of the gene. This is pure protein. Once this protein is synthesized, there are many things that can happen to this protein. It can be phosphorylation, acetylation, or methylation, or carbohydrate attached here, glycosylation, or lipid attachment can also happen. Even some other protein or peptide can be added here, and many more is under discovery right now. Why do we need so many different kinds of modification to the protein? We can think this way. Let's say we have one machine we made. Using one machine, sometimes you need to solve a different problem and different the enemy attack. Then you can generate maybe different derivatives or the different arms you can put up. Then you can generate a lot more diverse solutions. Rather than this method is maybe the more efficient than making out each individual different machines. So this is a similar situation in biological system, utilizing this post-translation modification. Using only one protein, but we can do many different things and many different functions, case by case. So those examples, as I said, it can be methylation or phosphorylation, glycosylation, or sulfation, or hydroxylation, or disulfide bond formation, or sometimes it can be lipid addition like prenylation or acid lipidations, and even sometimes adding protein or cleavage of those protein. Many possible modification can happen to the proteins. This is another view and another classification of those modification. Let's say if it is adding some chemical groups like a phosphorylation, acetylation, methylation. So these chemical groups can be removed too. So it's kind of an on and off situation. So this kind of modification can be reversible modification. Similar things like a lipid addition or glycosyl group addition, this kind of modification, or protein additions, these kind of additional the functional group can be reversible the reactions. But if the amino acid structure is changed, deamination or elimination type of chemistry happens, or cleavage happened, then usually this modification is irreversible modifications. Before I finish this lecture, I want to show you another movie to show you clearly where the post-translational modification happens in the cell.
Proteins move from the endoplasmic reticulum to the Golgi apparatus. Soluble proteins are carried inside vesicles, and membrane-bound proteins are carried in their membranes. Vesicles bud from the ER and move toward the Golgi cisphase. This vesicle is carrying soluble proteins, proteins not associated with the membrane. The vesicle fuses with the Golgi apparatus, delivering the proteins to the Golgi lumen. Another vesicle pinches off the ER, carrying proteins in its membrane. It also moves to the Golgi apparatus, and when its membrane fuses with the Golgi apparatus, the proteins it carries remain in the Golgi membrane. In reality, vesicles bearing membrane proteins may also carry soluble proteins that are recycled back to the ER if they are not ready for processing in the Golgi apparatus.
Okay, here is a second quiz for you. So there are many different types of post-translational modification, and I want you to go for and study at least some of these are the main modifications: phosphorylation, acetylation, methylation, and glycosylation and lipid modifications, and figure out which part of those protein is modified, means the functional group of the side chain of which amino acid is modified by these modifications. Here is another general other questions. If you have any questions from this lecture or any unclear part, then and also your suggestion you can put up on the poll ideally before the class. So hopefully by the Sunday night of this week, and send me your answers.